This demo consists of two parts. In the first, we will show how the shape alignment TRT models used for fish morphological feature extraction are trained and tested in the Visual Studio environment. And then we will see how the accelerated version of this method that we have developed in the context of the smartFish project works on a ZCU102 FPGA platform. In this video, we will show how we have adapted the deformable shape tracking package, DEST for short, for Windows and Visual Studio environment in order to train ensemble of regression tree models for fish shape alignment. These models are initially tested with applications on the DEST package in Visual Studio environment, and the best performing models that can be implemented with hardware acceleration are ported then to an FPGA platform like AMD Xilinx ZCU102. But first, let's see the tool we have developed called Landmark Annotator that is used to create the annotations of the training and test photographs needed for the development of an ERT model. Initially, I define how many landmarks I will use. Let's select 24. I upload the photo. And then, if I select New Annotation, I can start annotating. Okay, let's reset the system and load again this photo. But now I'm gonna load the already existing annotation. And here are the landmarks that I have already annotated. If I like, I could edit the annotation. I click a landmark and move it to a new position, for example. And then I could store the new annotation. Similarly, I would process all the photographs needed to create a large training and test set. Here we can see the environment of the ported DEST package in Visual Studio. As we can see, the solution consists of various projects, for example applications, needed to create and test an ERT model. For example, DEST train can be used to create a new ERT model. You can see here that I can give arguments that modify the number of trees, the cascade stage, the tree depth. All of these are parameters of the ensemble of regression tree models. Then I can define how the new model will be named. This is, for example, a name of a model that I want to train. Here it is the name. And the last parameter is the directory where the photographs along with their annotations exist. Then if I have already trained a model and I want to test the accuracy of this model against a specific annotated test set, I can use the DEST evaluate application. For example, here I defined which model I want to test and against which test set. In this directory, the test set exists where photographs along with their annotations made manually and serving as a ground truth exist. Then if I run this application, this project for example, I can see for this specific ERT model, these are the photographs loaded. And here we can see the normalized errors that have been estimated, as well as the error histogram. Then, if I set as a startup project the DEST Align project, we can use this project in order to test a single photograph. For example, here I define which model I want to use. This is the model I want to use. Other parameters, but then I defined the exact photograph that I want to test. When we start this project, we can see how the alignment is performed in each individual cascade stage. This is the fish and the first alignment made in the first cascade stage, the second, the third, fourth, the fifth, sixth, seventh, eighth, ninth, 10, and this is the final, which has the blue dots as the final position of the landmarks. As we can see here, we have tested a model with 26 landmarks. Now, I have changed the model to another model that is for just 4 landmarks, but I use the same photograph. Here are the results from the first cascade stage, the second, third, fourth, etc. And this is the final alignment. Finally, I will use a model that has been trained for photographs of fish outside the water and for 24 landmarks. And this is the photograph I'm going to test. Here are the results of the first cascade stage, the second, and this is the last. At this point, we see how fish morphological features are extracted using the hardware-accelerated environment on a ZCU102 development board. At the console at the right, we see the ZCU board booting from an SD card, while at the left, we will emulate through a FileZilla FTP client how the GPU-supported desktop sends fish image patches that are extracted from the camera video streams. When the boot is completed, we log in and then we mount the SD card and change directory. A Python script is started that detects if the special directory, in a special directory, if the FileZilla client or the desktop PC in the real environment has sent any photographs to analyze. Python script has started, and now we connect FileZilla with the FPGA boards. It is connected. And 3 photographs are sent in the target directory. We see that the Python script detected the arrival of these photographs, and now we'll call the application we have developed for fish shape alignment for each one of these photographs. In the target directory, we see that when a photograph is analyzed, a number of files are written named after the directory. Each directory is named after fish file name. For debug reasons, we store then not only the final list of coordinates of the landmarks, but also the intermediate ones that are produced from each cascade stage, and the fish photo annotated with the landmark coordinates produced at that stage. Here's the annotation of one photograph. Here is the final annotation of another one. In the version we have just viewed, processing each photograph is slow because the shape alignment application developed for the FPGA board is called for every photograph. This means that the ERT model parameters are reloaded each time. In order to see how fast the FPGA can perform shape alignment, we provide a faster version where the FPGA application is called only once when the image files have arrived. After boot, we have logged in and initialized the environment, and we also mounted the SD card and changed directory in order to start the new Python script. Python script has started, and now we connect FileZilla client. We send 6 photographs to the target directory of the FPGA board. The Python script detects the new photographs and starts the FPGA application. After loading the ERT model, the application processes the received photographs much faster. At the console at the right, we will see that the pure processing time for each photograph is just 0.01 seconds. This means 10 milliseconds, while all the 6 photographs required about 0.8 seconds, including the time needed to read the photographs and write the results back in the files. From FileZilla environment, we see that the results are visible for each photograph. Thank you for your attention.